Welcome and study with Tanzania Institute of Education.
Welcome, my lovely students, to our subject biology, and today's topic is nutrition. We are going to learn nutrition under a subtopic called the food test. Food test. Food test. Food test. This is the practical experiment which aim to detect the food contents in a given food sample. But since it is practical, today we are going to learn the theory of the practical of food test, aiming at finding what contents of food are found within a given food sample. Under food test, there are two types of food test. We have guided and unguided. We have guided practical and unguided practical. Under guided practical, there will be some instructions to allow you student to follow them, and through that instruction, one will identify the food sample and sometimes prepare a solution by yourself. The instruction may be, take a sample space, maybe an Irish potato, sweet potato, or carrot, and give you some procedures. Maybe the first procedure can be peel the Irish potato. Second procedure, cut it into small pieces. Third procedure can be grind it by using mortar and pestle. Fourth procedure can be add little amount of water. Another procedure, sieve to obtain the juice. And the last procedure can ask you to carry the practical test based on the solution obtained from the instructions which you were guided and until you prepare the solution. That is guided practical of food test. Another group or another category of food test is unguided. Unguided practical. In this unguided practical, you will be provided with the solution. The solution may be named maybe solution D, solution X, solution C. Your teacher or lab technician will prepare a solution from different food samples. I'm sure you know the groups of food. We have carbohydrate, we have protein, we have refuges, we have vitamins, we have water, and other types of food which comprise into a certain so-called food balanced diet, the foods that are used by human being in order to be healthy or to maintain your health. Somebody is supposed to eat a certain kind of balanced diet. Now, those groups of food to our secondary level, we are going only to test 3 types of food. Food number 1 to be tested will be carbohydrate. Carbohydrate. But this carbohydrate divided into some categories. Number 1 is starch. Number 2 is reducing sugar. And number 3 is non-reducing sugar. Reducing sugar. Another type of food which is going to be tested in the laboratory by secondary students is protein. Protein. And the last group of food that we normally test in the laboratories is lipid. Lipid. Lipid, I'm sure you know the components of it. It is fat, and oil. What is fat? Fat is lipid which is solid under room temperature or 25 degrees Celsius, while oil, a lipid which is liquid at 25 degrees Celsius, now forms the so-called lipid. These three types of food are tested in the laboratory, and there are some procedures that should be followed so that someone can end up with best results. And at the end of each test, a student will be supposed to write a food test report. And I want to teach you how to write a food test report. Starch, reducing sugar, non-reducing sugar, protein, and lipid. Write the food test report after the practical work. Some guidelines to be followed during report writing. There are several guidelines that should be followed during writing the food test report. Guideline number 1 is to specify the food sample. You specify the food sample, the solution given. In our case, we said our solution will be solution C. Now, solution C. The food sample is solution C. After specifying the food sample, in your food test report, consider the apparatus used. Consider the apparatus used. So you have to mention the apparatus. For example, a test tube, a white tile, beaker. A student should mention the apparatus used. So mention the apparatus. Mention the apparatus used. Number 3, specify volume. Volume of what? Specify the volume of food sample, the volume of solution C in our case. And it is recommended to use 2 centimeters cubed. It is recommended to use 2 centimeters cubed. Specify the volume used. Volume of what? Volume of either food sample and the volume of reagent or chemicals. In case of food sample, the recommended volume is 2 centimeters cubed in order to avoid the wastage of chemicals and the wastage of food sample. So you are advised to use 2 centimeters cubed of the said sample solution. Another procedure is to specify the correct procedure. Remember to mention the correct procedure. If the procedure is accompanied with the shaking, make sure that you write shaking. And if the procedure involves the process of boiling, make sure that the word boiling appeared in your report. And if the procedure needs you to make the material to shake, make sure that you will allow it to settle as per instruction. Allow it to settle for some times. There are some procedures which students should allow the material to settle for some times. Now, that is to abide. So to remember the correct procedure during report writing. Correct procedure. Remember to involve those things. Another is avoid the use of commanding language. During report writing, avoid the use of commanding language. Don't say put, add, shake. No. You have to use a polite language. Do not command during report writing. So avoid the use of commanding language, commanding terms. Use only polite terms. Another is, as it is a report, make sure that past tenses are used. Use past tense because it is a report on what you did. It is a report on what you did. So remember to use past tense. Remember to use the past tenses during report writing. Now, let's try to write the food test report. I will guide you how to write the food test report. What to do first, take your exercise book, divide it into phases like my board here. Divide your exercise book into 4 phases. The food tested, procedure, observation, and finally, inference. As we said, we have carbohydrate. And its 3 forms: starch, reducing sugar, and non-reducing sugar. We have protein and lipid. Let's start with the starch. The food tested here is the starch. What are you going to do during starch testing? You will take a test tube, add 2 centimeter cubed of the given sample solution, add 2 drops of the reagent, which is iodine solution. Then shake the mixture. 2 drops, then shake the mixture. You will see the color changes. You will see the color of the food sample changed into another color. Through that, you will identify the food sample contains starch or not. If the food will turn into blue-black, it implies that the said solution has starch. But if the food sample, the solution, retain the color of iodine solution, which is brown, it implies that there is no starch in the said sample solution. Now, how to write it? You will say, in a test tube containing 2 centimeter cubed of solution C, 2 drops of iodine solution were added. 2 drops of iodine solution were added, then shaken. Observe the color change. If the food sample contains starch, the color of the solution will turn into blue. Now, the combination between food sample and iodine solution will form a mixture. Now, the mixture turned into blue-black. Implies that starch was present. Starch was present. That's first phase, if the food sample contains starch. What if there is no starch in the food sample? The procedure remains the same, but the observation here, the mixture will not turn into blue-black, but never write the mixture did not turn. Write what you have observed. Here, the mixture retained the brown color of iodine solution. Iodine solution means starch was absent. This means starch was absent. Never write the solution did not turn. The solution turned from the said color of the reagent. Maybe it was white color of solution C. After addition of 2 drops of iodine solution, the food sample turned into brown color of iodine solution. Though your expectation was the food to turn into blue-black, but not turn into blue-black. What was the color formed? Now you have to specify that color formed, which is now brown. Next test is reducing sugar. Reducing sugar. The same procedure, those 5 elements in a test tube contain 2 centimeter cubed of solution C. 2 centimeter cubed of Benedict's solution. We have a certain solution called the Benedict's solution, blue in color, was added, then boiled. Within the food sample of 2 centimeter cubed, 2 centimeter cubed, another 2 centimeter cubed of Benedict's solution was added and the mixture was boiled. You will observe the color changes when boiling. The first minute, you will observe the color changes. The color will change from blue to green to yellow, and finally, the color, a series of color change will be observed from blue to green to yellow, and finally orange. Finally orange. This implies that the food content contained reducing sugar. So reducing sugar was present. Reducing sugar was present. As I have said, the Benedict solution is blue in color. If there is no concentration of reducing sugar to the said sample solution, the mixture will retain the color of Benedict solution. Therefore, the report should appear like this. The mixture retained the blue color of Benedict's solution. Benedict's solution. This means reducing sugar was absent. Reducing sugar was absent. That was the test of starch and reducing sugar. Next is the test of non-reducing sugar. Let's see how are we going to write a report of non-reducing sugar, a test of non-reducing sugar. You remember the procedure? Specify the test tube used, the apparatus used, specify the volume, remember to mention the name of the solution used, remember the correct procedure if it's to shake or if it's to boil. Now it's non-reducing sugar. Non-reducing sugar. Here, the same introduction. In a test tube containing 2 centimeter cubed of solution C, 1 centimeter cubed of dilute hydrochloric acid. Hydrochloric acid was added. Then the content boiled, and cooled. After cooling the components, 1 centimeter cubed of sodium hydroxide solution sodium hydroxide solution was added for neutralization. Then 2 centimeter cubed of Benedict's solution was added, and the content was reboiled. In a test tube, your test tube is here, 2 centimeter cubed of food sample, 1 centimeter cubed of HCl, hydrochloric acid. Here is solution C. Then boil. After these two, you boil the content. Then allow them to cool. After cooling, 1 centimeter cubed of sodium hydroxide was added. Now, why HCl? Why sodium hydroxide? This is non-reducing sugar. The first procedure is to convert, because there is no direct test of non-reducing sugar in the laboratory. Therefore, you have to convert it from non-reducing sugar to reducing sugar first. Now, the process of converting uses the so-called hydrochloric acid. Therefore, your non-reducing sugar reducing. After addition of 1 centimeter cubed, you will boil it in order to catalyze, to accelerate the reaction. The reaction will to convert the non-reducing sugar into reducing sugar. Within the test tube, the non-reducing sugar will be not there, but the content will contain only reducing sugar and HCl remained. You are supposed to remove this HCl because what we want is only reducing sugar, which was converted from non-reducing sugar by using this HCl. So you will add 1 centimeter cubed again of sodium hydroxide in order to neutralize this acid. A combination between acid, HCl, and sodium hydroxide is called the neutralization reaction, because any reaction between acid and base produces salt and water only. Therefore, you will remain with the only reducing sugar. Then you will proceed with the normal procedures of testing reducing sugar, and the results will be as follows. You know, if we have already converted from non-reducing sugar to reducing sugar, you have already your reducing sugar. Now, the addition of Benedict's solution is just a normal test of reducing sugar. You observe a series of color change from blue, the color of Benedict's solution, to green, to yellow, and to yellow, orange, and finally, if the concentration of non-reducing sugar is high, you will end up with finally the color of brick red, and finally brick ready. You will observe the series of color changes during reboiling. You will observe the color of series changes. Here implies that non-reducing sugar was present. If there were no non-reducing sugar in the food content, the mixture retained the blue color of Benedict's solution. This implies that non-reducing sugar was absent. Non-reducing sugar was absent. This is the test of carbohydrate under its 3 states: the starch, reducing sugar, and non-reducing sugar. Another test is the test of protein, food test under protein. The procedure: protein. During protein test, the point to be observed remain the same. Specify the apparatus, mention the food sample, specify the volume used, make sure that you observe the correct procedure, and don't use commanding language during report writing. Here is protein, the test of protein. In a test tube containing 2 centimeter cubed of solution C. In a test tube containing solution C. Two centimeter cubed of sodium hydroxide solution was added. Sodium hydroxide solution was added, followed by addition of 2 drops of copper 2 sulfate solution. Copper 2 sulfate solution. Then the mixture was shaken. After each drop, you have your test tube, 2 centimeter cubed of food sample, 2 centimeter cubed of sodium hydroxide solution, and 2 drops of copper 2 sulfate solution. Then shake after each drop. This means when the first drop enters in the mixture of food sample and sodium hydroxide, shake the contents. The second drop again, shake the contents. You will observe the changes of colors in the mixture. If the food contains protein, the color of the mixture will change into purple. Other people say it is violet. It can either be purple or violet. But during report writing, remember to write only single color, purple, or you may write violet. You select one of the two. I prefer to write purple. So the mixture turned into purple color. This means protein was present. Protein was present. And if there were no any components of protein in the solution provided, means the color of copper 2 sulfate solution is blue. And this sodium hydroxide just colorless. Therefore, the color of copper 2 sulfate solution will dominate the mixture. So the mixture turned or retained the blue color of copper 2 sulfate solution. Copper 2 sulfate solution. This implies protein was absent. Protein was absent. The last food to be tested in our sample is lipid. During this practical procedure, you have to observe 2 things. Number 1, the procedure of shaking the mixture vigorously after addition of the reagent to the food sample. you have to shake it vigorously in order to mix up the concentration of food sample and either lipid, which is either fat or oil. Then you will let the mixture to settle in a test tube racker for 5 minutes in order to allow the clear separation between the lipid concentration and the solution C contents. And this will give you a clear picture. If the food sample contains lipid, you will observe a red ring formed on top of solution C. You have your test tube here, the food content here, which is 2 centimeter cubed, then 3 drops of Sudan 3 solution. When you shake this content vigorously and let them to settle for 5 minutes, you will observe that there will be a formation of red ring here on top of solution C. If a red ring will be formed here, it implies that the food contains lipid. But if there will be a clear mixture between Sudan 3 and the solution C, it will form a red solution, means there is no lipid in the food sample. Now, to write this procedure, you have to start with the lipid as usual to this side in a test tube containing 2 centimeter cubed of solution C, 3 drops of Sudan 3 solution were added, then shaken vigorously. And allowed to settle for 5 minutes. A red ring layer was observed on top of solution C. This means lipid was present. Lipid was present because the lipid has an ability of catching the components of Sudan 3. If there is no lipid, means the mixture, the mixture acquired the red color of Sudan 3, solution. This means lipid was absent. This is how someone can perform an experiment of food test and also writing a food test report. Before I conclude my lesson, I leave you with a question. You have to write it down. Note this. Assume you are provided with a solution prepared by using 1 egg and porridge. You have a solution prepared by 1 egg and a porridge. That mixture, term it as solution D. Perform a food test procedure, then write a food test report. Assume you have a solution prepared by using egg and porridge. Perform a food test to identify the food contents which are in that food provided, and let it be solution D. You were with me, Madam Consolata Blas. Till another time. Bye.